Dear colleagues and students, authorities, ladies and gentlemen, it is my immense pleasure and honor to give this laudatio for Professor Katalin Karikó. To quote a title from the New York Times, Dr. Karikó helped shield the world from coronavirus. This is because she contributed to develop the technology of mRNA COVID-19 vaccines. Let me indeed give you some numbers to highlight the impact of her findings. In the first 6 months of 2021, it is estimated that mRNA COVID-19 vaccines have saved 280,000 lives and more than 1 million hospitalizations, only in the US, without mentioning the avoidance of the economical and psychological consequences of prolonged lockdowns. But what is an mRNA vaccine? SARS-CoV-2 infects human cells in the nasal and oral cavity. You can see here, these are the cells that are taken up by the virus. Wait a second. Oops. Okay. So the virus enters via these cells, which are epithelial cells, in the oral and nasal cavity, and it binds to one receptor on the cell, which is called ACE2, via a protein which is called spike. You have all heard about it. And then this allows the virus to enter the cells, and then the infection starts. So this interaction between the spike protein on the surface of the virus and the ACE2 protein on the cell membrane of the host cell allows the interaction and allows the virus to penetrate. So the vaccine, what it does is to induce the production of these antibodies which block this interaction, and in this way they block the virus to enter the cell. You can see here in this animation how it works.
Coronavirus. Like many viruses, it uses a protein on its surface to attach to and enter our cells. Antibodies that fit onto this protein can block the virus from attaching. Coronavirus mRNA vaccines teach our immune system to make these antibodies.
So you have seen why it's shielding, because it's really covering the virus and not allowing the virus to enter. So we can have 3 different types of vaccines. Those that are a DNA, like the AstraZeneca one, the RNA, like the Pfizer and Moderna, and the spike protein, like the Novavax. We still don't have it in Italy now. So it's like having a mold, like the DNA, a cast, the RNA, and the protein itself, the spike protein. But what you want to do is to make this protein, which is the spike protein, because you want to make antibodies to this protein. So then the mRNA vaccine is made of 2 components. One is the liposome, which carries the RNA, which is inside within the cell, and it protects from degradation. Once it is inside the cell, this mRNA is carrying the information to make the spike protein, and the spike protein, as I told you, is the one that you want to induce the immune response to. Here you can see in our experience on healthcare workers, this vaccine is very, very powerful. You can see here that already 1 dose of vaccine is capable of inducing an immune response in all of the subjects except for 1. But then you need 2 doses to have achieved maximal induction of these antibodies, and they last at least 3 months. You can see here that in naturally infected subjects, so those that have a COVID-19 history, you need just 1 dose in order to have the same type of immune responses, 2 doses in naive subjects. So this allowed us to say that in COVID-19 experienced people, you just needed 1 dose, and we have saved a lot of doses for individuals that really needed them. Okay. So what makes mRNA vaccines so special in comparison to other vaccines, and what this discovery from Dr. Katalin Karikó is important? So despite what many people think, it took more than 30 years to make the mRNA technology to work. So when people argue that the mRNA technology is too young and that it's not safe, they should talk to Dr. Karikó. Further, before the COVID-19 vaccines, the mRNA-based biologics and vaccines had been tested in clinical trials for several years, mostly by Dr. Caricot and her colleagues, in various applications demonstrating safety. But why did it take so long to develop effective RNA-based vaccines? First, the stability. So RNA, as it is found in nature, is quickly degraded, which makes it safer but also reduces efficacy. And second, it could activate immune cells and induce inflammation. Dr. Caricot studied both, allowing to solve these problems. After working as an undergraduate on lipids, Dr. Caricot started working on mRNA by serendipity, as she accepted a PhD position in Geno Thomas' lab in Hungary. Here she realized that mRNA could be used as antivirals. When she went to the US from Hungary, she continued working on this concept at Temple University in Pennsylvania. She then tried to use the RNA technology to deliver proteins of interest within the cell, and in particular, urokinase receptor to treat cardiovascular diseases, and cytokines to treat cancer. So these studies are still ongoing, showing the potential of RNA technology beyond vaccine development. When Dr. Caricot was struggling to make things work, many researchers and companies thought that mRNA was too unstable and expensive to be used as a drug or a vaccine. Even Malone and Molien, who were pioneering the mRNA technology for drug development, gave up and moved into DNA-based vaccines, which were more stable. However, Dr. Caricot was not discouraged and continued to work on this complex subject because she believed in it. Quoting her words, "People felt sorry for me working on RNA, as it is boring and so difficult to work with. But as many scientists would agree with me now, she took what happened to be an uphill path, but which proved to be the right one. This is a teaching for every student and researchers in this room, so don't give up." It was in 2005 when the mRNA technology developed by Kathleen Caricot and her immunologist colleague Drew Weissman, both at the University of Pennsylvania, in Philadelphia, became useful for vaccine development. By substituting uridine, one of mRNA nucleotides, with an analog called pseudouridine, allowed to stop the body identifying the mRNA as a danger and allowed increasing of 10 times the amount of protein which was produced. So you can see here, this is the major advantages of what Dr. Caricot put in place. So first of all, you have the lipid molecules inside of which you can find the RNA, and there is a substitution of the uridine into pseudouridine, and this allows this RNA to be more stable and also to be less immunogenic. It took, however, years for Kathy to convince the scientific community and investors that this modification would move forward the mRNA vaccine field. Her funding proposals were rejected, but she never gave up. 8 years later, in 2013, she went to give a seminar in Mainz, Germany, on the way back to Hungary from Lucerne, during one of her daughter's rowing competitions, because you should know that Kathleen is not the only star in her family. Her daughter, Susan Francia, is a pluri-Olympic and world rowing championship, so it's a very talented family. In Mainz, Kathleen met Ugur Sain, the CEO of BioNTech, and started working for BioNTech as a vice president, quite a big change. Both Moderna and BioNTech vaccine contain modified mRNA according to the findings of Kathy and Drew. This explains why they are the best vaccines in use. CureVac is not using the same technology. I had the chance to attend by remote, the beauty of being in a new world, a seminar given by Dr. Caricot at Cambridge University. Besides having really appreciated her sense of humor, after the seminar, she said something very important. People working in science work for the pleasure of making new discoveries, to understand how biology works. It is curiosity that drives our work, no matter how difficult it is to reach achievements. A publication in top-notch journals, Nature, Science, should not be what drives our science. Solving little problems and sharing of the data is what makes scientists successful. Dr. Caricot has remained very humble, even after being finally recognized for her fantastic achievements and for having saved thousands of lives, which is the dream of every scientist in biomedical translational research. I hope I will do it someday. She's also a role model for females who want to get into STEM subjects. She says that if you want to be a good scientist, you should find the right husband who is supportive, does not complain about long working hours, and learns how to cook, fundamental. I wouldn't agree more, but I think that what made Katalin very successful is resilience and courage. She left Hungary in 1985 with around $1,200 hidden in a teddy bear, a dream, a passion for science, and a 2-year-old little girl. She had several stops in her career, but never gave up on her mission and made curiosity drive her science. The mRNA technology that Katalin contributed to establish has far-reaching potential and applications in other fields, including cancer vaccines and therapy, from which it actually started, and therapeutic potential for several other disorders. It is a multifaceted technology with great potential. As a scientist, woman, and mom, I'm very grateful to Katalin for her resilience in science, for driving us out of the pandemic. Thank you, Katalin.